All right, good afternoon. My name is Roarick Schollmeyer, and I am presenting one of the projects that I've had the special privilege of helping research on. This research looked at treating brain tumors, specifically glioblastomas with nanoparticles complexed with polymers and siRNA, in an effort to see GPX4 knockdown and subsequently reducing radioresistance. Glioblastomas are among the most common primary brain tumors in adults and are notoriously lethal. Only about 2% of patients with glioblastoma are able to survive 5 years past the date of diagnosis. Current treatments for glioblastoma include the use of chemotherapeutics or radiotherapy. This is a problem because glioblastoma has the ability to develop resistance to these treatments and thus make rehabilitation nearly impossible. Studies have shown an association between high mesenchymal cell state in glioblastoma cell lines with the ability to develop resistance to multiple treatments. To make matters worse, the delivery of drugs to the glioblastoma is greatly impeded by the blood-brain barrier, which only allows certain small molecule drugs to cross due to the epithelial-like tight junctions within the brain capillary endothelium. Now, this therapy-resistant high mesenchymal cell state in glioblastoma has been shown to depend on a druggable lipid peroxidase pathway, which offers protection from ferroptosis, a non-apoptotic form of cell death induced by the buildup of toxic lipid peroxides. Glutathione peroxidase, or more commonly known as GPX4, is an enzyme that dissipates lipid peroxides. When present, there will not be a sufficient level of lipid peroxides to induce ferroptosis of the glioblastoma cells, which we believe helps give rise to this high mesenchymal cell state. So we plan on inducing deliberate knockdown of GPX4 via siRNA. However, we still run into the issue of the blood-brain barrier, making it very hard to get siRNA directly to the glioblastoma cells. So this is where the nanoparticles come in. Some nanoparticles have the ability to cross the blood-brain barrier and precisely target tumor regions, which potentially makes them very useful for delivering medication to glioblastomas. The nanoparticle that we're using in this lab is comprised of an iron oxide core that has been functionalized with polymers, which allow for the attachment of various drugs and cellular targeting agents for the targeted delivery of the drug. As shown in my diagram, the negative charge of the siRNA complexes to the positively charged primary amine groups that are present on the outer CP-PEI layer. Now, please keep in mind that the diagram is most definitely not drawn to scale. In reality, the iron oxide core would be a lot larger, but you see the inner two layers, the cross-linked silane and the PEG. Those are polymers that are present to increase stability of the particle itself. Also pictured in the diagram is complex chlorotoxin onto the nanoparticle, which targets receptors that are overexpressed in glioblastoma, which means that we can hopefully selectively target glioblastoma cells. To determine upregulation of GPX4 within glioblastoma cells, we plated a U118 cell line comprised of human glioblastoma cells and incubated them under hypoxia for 11 days. The hypoxic environment was kept in order to mimic the microenvironment that would promote glioblastoma cell invasion or spreading into healthy brain tissue, which is the main cause of death in glioblastoma patients. Cell samples were collected on specific days and were frozen down for RNA extraction, followed by cDNA synthesis and qPCR analysis. Now, in both of these graphs, you'll find that the incubation time of the cells are placed on of the cells placed in hypoxia is on the x-axis, and on the y-axis, you'll find relative normalized expression of the respective target tested. And after running qPCR, we were able to confirm that there was a trend of increased expression of the GPX4 pathway under prolonged hypoxia. I should also include that, I also chose to include the qPCR results for NCAD-herin, which is one of the epithelial mesenchymal transition markers that has been shown to increase in expression when exposed to a hypoxic environment. We also tested EMT markers, snail, ecadherin, vimentin, twist, and fibronectin, and all these markers were expressed in higher levels with increased hypoxia exposure. So with this in mind, we took the project to the next level by running an experiment where we treated glioblastoma cells with siRNA complex nanoparticles with the goal of seeing GPX4 knockdown. Now, unfortunately, I actually did not have time to see this experiment all the way through and actually missed the ability to collect the data by 1 day, so I'm a little sad about that. But if the results for this experiment are promising and we gather successful GPX4 knockdown results, then the next steps for this project are to establish comparative radiation survival curves in an in vitro setting before moving forward into an in vivo testing. It is our hope that nanoparticle-mediated knockdown of GPX4 proves to be an effective way to lower glioblastoma cells' ability to develop resistance to traditional treatment modalities. So I'd like to now acknowledge Dr. and Ms. Ellenbogen for giving me this amazing opportunity to participate in this program. And I also want to thank Zach, Stefan, Guan Yu Lin, and Haley Locks for their generous patience and invaluable guidance throughout this program and my research experience. Julie and Sylvia for their marvelous support and prowess to smoothly coordinate this program. The remainder of all UW staff, donors, and residents for facilitating my journey throughout this program. And lastly, all of my colleagues for their strong ambition to be great. You guys have encouraged me to be the best that I can be, and this program has been just an absolute surreal experience that has made for a sublime summer. Thank you all.